So step one in this case is we need something to break down, and we're going to break down glucose. That's going to be C6H12O6. Okay, and glucose is going to be broken down into how many pyruvate?
2.
Okay, that process is called glycolysis. And it's going to yield us pyruvate. Specifically, it's going to yield us 2 pyruvate. Okay? What are we going to make in glycolysis? What? 2 ATP, and what else? What else gets made? NADH. So NADH, specifically 2 NADH. And 2 ATP. Okay? Then our pyruvate is going to go on into what site or what step? Not yet. Prep steps. How many times are we going to do the prep steps? Twice. 2 pyruvates yields 2 prep steps. Okay? What do the prep steps produce for us? CO2. And what else? Acetyl coenzyme A. How many acetyl-CoAs? And what else does it produce? Two NADH. Okay. Where does the acetyl-CoA go? Krebs cycle. How many times do we go through Krebs? Twice. Once per acetyl-CoA. And what does the Krebs cycle produce for us? What? Yeah, it's going to use H2O. What is it going to produce? Lots of stuff gets produced. What's anything that gets produced? NADH, yeah. Anybody know how much? 6 NADH. And anything else? FADH2. How much of that? Oops.
4 C.
Yep, that's a waste, so we're not going to include it, but what else? One other good thing. Yeah, GTP. I heard somebody say it. And we can just think of this GTP as the same as ATP. Okay, how many of that do we make? 2 of them. Okay, so now there's nothing else that got exported from the Krebs cycle that we can do anything else with, right? So we made acetyl, or you made CoA again, so go back here. But for the most part, we're done with this glucose molecule. It's all used up. So then we use all of the product to the stuff that we've done so far. And this all goes in to the electron transport chain. Right? And we said that the electron transport chain is going to make hydrogen ion gradient. It's going to be able to pump 10 H plus ions per NADH. And it's going to be able to pump 6 H plus ions per FADH2. And then it's going to convert that to 1 ATP per roughly 3.5 H plus ions. We said we could do some simple math here, and we could say that if we have 10 NADH, that we can divide that by 1 NADH per 10 H plus ions. And that's going to give us 100 H plus. Similarly, we have 2 FADH, or FADH2 rather. And we can say 1 FADH2 is going to pump 6 H plus ions. And that leaves us with 12 from that. And so 100 plus 12 is 112. So we have 112 H plus ions. We said that 3.5 H plus ions is going to yield us a single ATP. And therefore we can say that a single ATP or 112 H plus ions will make us 32 ATP. Okay, so this 32 ATP is created through the process of what's called oxidative phosphorylation. Right? So overall, we've got 2 ATP from glycolysis plus 2 ATP from Krebs plus 32 ATP from ETC. The blue ones on here, this is substrate level phosphorylation. The red one, that's oxidative. And so overall, how much do we make? 36 ATP total. Right? Okay. So, in your book, it says 36 to 38 ATP, right? Now, what's the difference? The difference is, this is the way that I teach this because everything works out nice and clean and it's pretty to look at. All the numbers are round, right? It's not really what happens. This is a little disingenuous because these numbers up here are fudged a little bit. This 10 H plus ions per NADH and 6 H plus ions per FADH2. That's a little bit fudged, and then this one is fudged a little more than that. So this is more like 3.625 H plus ions, right, per ATP. And so you end up with a number that's much closer to 38, right? But that doesn't explain why there's a difference. The difference, and really the differences in the numbers, I don't really care if you know about that. It doesn't matter at all to me. If you just learn these numbers will last you as long as you're ever going to want to go, probably. right? I would say even if you went to medical school, you're not going to have to know any different numbers than this, right? Unless you're like going into specifically molecular biology or something like that. That, you might be like, well, no, it's actually 3.625 H plus ions per ATP. Then they might care, but for the most part, nobody's going to care the difference here. Why is there a difference, though, between 36 and 38? That's probably important information. So the difference comes from eukaryotic and prokaryotic cells. So eukaryotic cells are going to produce closer to 36 ATP. Prokaryotic cells are going to produce closer to 38 ATP. And the reason for that is that glycolysis happens in the cytoplasm and produces 2-pyruvate. Okay? So there's a fairly low concentration of pyruvate in the cytoplasm, and that pyruvate has to go into the mitochondria. But that's where all the pyruvate's going. So the mitochondria has a pretty high concentration of pyruvate. So in order to get the pyruvate into the mitochondria, it has to be actively transported in there. Specifically, it has to be actively transported with 1 ATP per membrane that it has to go through. So, how many membranes does it have to go through to get to the mitochondrial matrix? 2. It has to go through the outer membrane of the mitochondria, and it has to go through the inner membrane of the mitochondria, right? So 2 ATP gets used to get that pyruvate inside, and therefore 38 ATP gets knocked down to 36 ATP in eukaryotes. But in prokaryotes, they don't have mitochondria, and therefore that's not a thing, right? So that's where the discrepancy comes from. But if you just learn it like this, I promise this is plenty good for anything that you're going to need it for, right? All right. (36 ATP per glucose. Anybody in here on a pure glucose diet? Just scoop it up, eat it. It's terrible. It would be a horrible thing for you. You would die in not very much time, right? Not to mention you'd have to be constantly eating glucose, because it's metabolized really quickly. So you'd have to have like a nice steady stream of it, where you're like taking a scoopful every 45 seconds or something, right? And that would be terrible. So that's obviously not where we're getting the majority of our metabolism from. It's coming from other places. So we got to learn where else it can come from. So other metabolisms. All right, so we learned this already. What thing has the most energy per molecule? Fats. So we'll start there with fats. Specifically, we're going to talk about triglycerides. They call them triglycerides because, why do they call them triglycerides? 3 something. 3 what? Nope. 3 fatty acid chains on what? Attached to 1 glycerol. Okay? So, glycerol looks like this. It's 3 carbon chain. And then attached to that, we've got fatty acid tail. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm just doing 10 carbons here. It's actually pretty small for a fatty acid. Generally speaking, we expect these to be somewhere between 15 and 30 carbons per tail. So even more energy dense than this one that we're talking about. But this is going to be fine for now. Okay? So this thing needs to get metabolized. It's really big, and so it's not going to fit nicely into any of those enzymes that we've looked at thus far. So it's got 2 parts to it. This part up in the front is called glycerol. And in the back, we've got fatty acid chains. We're going to assume, just for simplicity's sake, that these are saturated fatty acid chains. I did some some scrap paper work here and determine that the formula for this fatty acid that I've done, this example, is C36H38O6. There's some oxygens in glycerol here, and there's an oxygen in this ester bond too. So that's where those 6 oxygens come from. All right, so keep that in mind. Let's talk about where these things are going to go. So glycerol is a 3-carbon chain. So we're going to need to process glycerol like it's a 3-carbon chain. What 3-carbon chain gets processed in Krebs cycle, or in prep steps rather? Pyruvate. So the very first thing that we need to do is we need to convert glycerol into pyruvate. Okay? Once we've converted glycerol into pyruvate, we can send pyruvate into the prep steps. How many pyruvates are we going to make? How many? Here's glycerol. So it's just one, right? Because 1 3-carbon chain glycerol turns into 1 pyruvate. So we just do the prep steps once. Then the prep steps goes into acetyl-CoA. And then the acetyl-CoA goes where? Krebs, right? Okay? And how many times are we going to do the Krebs cycle? 1 time. Okay? So that's going to make us a little bit of stuff that we need, NADH and FADH2. But the big thing is going to come from all these fatty acid chains. And so the way that this is going to get processed is these fatty acid chains are going to get chopped up into 2 carbon groups. And every single one of these 2 carbon groups is going to essentially have the formula C2H4. And those things are going to add an OH group to them. Okay? And as it adds an OH group to them, it's going to become an acetyl group. Okay? So when you take these things and you add an acetyl group to them, what molecule do you think we're going to make with that? What molecule can we make with an acetyl group? Acetyl coenzyme A. Okay. This process is very well understood. It's called beta oxidation. That's that process there. And beta oxidation is going to produce 1 NADH and 1 FADH2 per acetyl-CoA. In other words, we're going to take these groups here, we're going to oxidize them in beta oxidation, and we're going to take away 4 electrons. 2 of the electrons get added to NADH, 2 of the electrons get added to FADH2. NADH and FADH2. Okay? Where does the acetyl-CoA go? Krebs. All right. So for this fatty acid, or this triglyceride, how many acetyl-CoAs did I produce? 15. So how many times am I going to do the Krebs cycle? 15 times. All right, so let's do the maths on this. Over here, we did the prep steps, right? What did the prep steps give us? They give us anything we need? What do the prep steps produce? What? Acetyl-CoA, and what else? NADH. Yeah, so we're going to make 1 NADH from this. Okay. Krebs cycle makes how much?
6.
6 for 2 cycles. How many cycles do we get to go through here? Just 1 cycle. So we're going to go 3 NADH. How much FADH2? Just 1 in this case, because we're only going to go through it once. And how many GTP or ATP we'll call it? Just 1. I'm just going to put ATP. I'll put it in parentheses so you know that it's actually GTP, but okay. So when we produce these 15 acetyl-CoA through beta oxidation, we're going to make 1 NADH and 1 FADH2 per. Right? So that means that we're going to make 15 NADH and 15 FADH2. Then we're going to go through the Krebs cycle 15 times. So 15 times 3 is 45 NADH that get made there. 15 times 1 is 15 FADH2. And then 15 times 1 is 15 of these GTP things that we're just going to call ATP. Okay? So then we got to go do some maths. So we got 15 plus 45, that's 60, plus 3 plus 1, that's 64 total NADH. And we said that 1 NADH could pump 10 electrons, or sorry, 10 H plus ions. And so that means that we have 640 H plus ions pumped there. All right. Then we had FADH2, so we had 15 plus 15 is 30 plus 1 is 31. So 31 FADH2. One FADH2 makes 6 H plus ions on the gradient. 31 times 6 is 186 H plus ions. So 640 plus 186, that's 726, 826. 826 H plus ions. We said that 3.5 H plus ions produces 1 ATP. That one I can't do in my head. Consult the calculator. So 826 divided by 3.5 is 236 ATP. This 236 ATP is through oxidative phosphorylation. And then we also had some more. We've got 15 ATP here and 1 ATP here, so plus 16 from substrate level. And so that gives us 252 ATP. That's a lot, right? How many did 1 molecule of glucose make us? 36 versus 252. Fats are pretty energy dense. Now, It's not as energy dense as it seems, right? So some quick math here. Again, on my paper, I've done this math. You just kind of have to trust me, okay? So, and you don't have to write this down, but it's interesting to know. So, oh, bummer. This tape. Okay. All right. So if you looked at the energy density, right? So one molecule of glucose has a molecular weight of 180.18, and it makes 36 ATP. So if we took 36 ATP and we divided by 180.18 AMU or whatever, right? It's really grams per mole, but whatever. Okay, so that's for glucose. And then for this fat, and remember it's going to be different based on how long the fatty acid tails are. But I came up with, it was, how many ATP do we make? 256? No, 252? 252 ATP per 566.74 AMU, right? And so the way I got that was I took this formula up here and I just calculated the molecular weight of that formula, right? So if we do this, we do 36 divided by 180.18, we get we'll just call it 0.2, right? Pretty close. Yeah. And then this one was 252 divided by 566.74, and that one's like 0.44. So over twice as dense, but still not like as much as it seemed, right? So it seemed like it was like 100 times as dense or something like that, but it's not. It's twice as dense, or 10 times more, I guess. If it's like twice, right? Right. Okay. So do you only eat fats and sugars? I would like to only eat fats and sugars, but that would work out very poorly for me. So what else do we eat? I guess I couldn't eat chicken if I didn't have protein, so I'd take that back. I love chicken. Proteins. Your body can't metabolize proteins. It's got to break them down first. What do proteins break down into? Amino acids. Okay? So amino acids are going to be metabolized in a bunch of different ways. You start out with amino acids. The first thing that happens is they have to get their amine group broken off of them. Okay? Their amine group is a nitrogen-containing group, and that nitrogen-containing group gets turned into a nitrogenous waste product called urea, which is in urine, right? It's the number one molecule content in urine other than water. So that means that people who have high protein diets will have high urea content, and that's why they tell you if you are having a high protein diet, you need to have a lot of water also, because the water is going to dilute the urea so that you don't get kidney stones that are made up of urea. Okay? Which is bad. You don't want kidney stones. So the rest of them are going to depend. So it can go in a couple of different places. It can go into pyruvate, or it can go into acetyl-CoA, or it can go into what's called succinate. If you remember, succinate was the 4-carbon chain at the bottom of the Krebs cycle. Okay? So what determines what it makes, whether it makes pyruvate or acetyl-CoA or succinate? It determines that based on the R group. So the molecule produced depends on the amino acids R group. Right? So the R group is what makes the amino acid unique. One amino acid is different from another one based on its R group. So there's 20 different R groups, and those 20 different R groups are going to fall into one of these three categories: pyruvate, acetyl-CoA, or succinate. Okay? So overall, you know that cellular respiration goes from glycolysis into Prep steps, into Krebs, and then into ETC. And if you have a carbohydrate, they're going to go directly into glycolysis, right? If you have fats, fats are going to enter in two places. The majority of the fat, the fatty acid tail, is going to enter at the beginning of the Krebs cycle after it gets made into acetyl-CoA, right? But the glycerol part of it is going to go into the beginning of the Prep steps because it's going to get made into pyruvate, right? And then proteins are going to go in a couple different places. They can either enter halfway through the Krebs cycle as succinate. They can enter at the beginning of the Krebs cycle as acetyl coenzyme A, or they can enter at the beginning of the Prep steps as pyruvate. Questions on that? All right.